All right, looks like we are live here on Standing for Truth. Welcome everybody to the show. It is a privilege to have Jamie from the Studio 215 official YouTube channel. Tonight we are going to be focusing on single cell to multi-cell evolution. Jamie has an excellent presentation for us specifically on this topic. And I know some people in the audience, they were thinking probably that this is supposed to be a debate, Jamie versus Taylor, or Snake was right. And so we have rescheduled that one probably for next week or the week after. We do have a lot of events coming up for you. Taylor had an emergency come up, and so we hope everything is well and everything is going to be okay for him. But we will most definitely be getting that debate. Jamie is also going to be here next week. He's got another debate with James W. So James W. and Jamie, and they're going to be debating the classic creation versus evolution. So I'm pumped for this, which means we'll probably be rescheduling the Taylor debate shortly after this one. So we are keeping Jamie nice and busy. And so what I want to say, though, is we are going to make this interactive at the same time. And so Jamie, he's going to be giving about a 30-minute to an hour presentation on this topic. Now, this is an argument. There's several papers that those in the evolutionary community have been utilizing, trying to say that we have evidence in real time for multi-cell evolution, multicellularity in real time, basically. Jamie, I want to thank him for doing so much study on this topic. And we want to make this the go-to video for anybody who's confronted with that argument. And so they can check out this video. It's going to be a solid presentation. It's going to be very comprehensive on this topic. And it really is great that Jamie's done so much study on this. And so after his presentation, 30 minutes to 60 minutes, I've got the link posted in the live chat, the join link for StreamYard. So any evolutionist, anybody of a differing view or opinion than Jamie on this, click the link and we will allow you to join and have some interaction with Jamie. So we'll make that fun, and we'll do that around the hour mark, and then we'll kind of cap this at 2 hours. So Jamie, before you get into the presentation though, brother, great work over on your channel. I'm loving the Bible studies. I'm really appreciating the content you're putting out. Really enjoying your debates. I'm glad that we got some more for you. And I know you've got a challenge out there. Anybody who wants to debate this topic or anything in the creation versus evolution world, reach out and we'll set that up. So Jamie, tell us a little bit about yourself. How have you been as well? And tell us a bit about your channel.
Yeah, for those that don't know me, my name is Jamie. Well, obviously you said it like 15 times already. But yeah, I run the Studio 215 official YouTube channel. I'm also building a website and doing a lot of other projects as well. I've recently jumped into the documentary phase of my studio. So I've got a couple documentaries that I'm working on. One documentary on the Ron Wyatt discoveries, one documentary on giants. It seems like that's going to be another one that I have to add to my list. Another documentary on the King James. I'm probably going to end up doing a documentary on Claiming Demonis. So, I mean, I got a lot of projects in the works. So, you know, go check out my channel, hit that subscribe button. There will be a lot of content moving forward. I'm also doing those Bible studies, like you said. So every week we do a live version of the Bible studies on my Discord channel. So you can go to the Discord channel if you want to join the live version for the fellowship. I also will take, if anyone has something they want to input to that, I'll consider that for adding notes into my presentation. And then the following week, we actually post a recorded version of it. So for anyone that's looking for fellowship out there, feel free to join my Discord channel because that's what we do over there.
Awesome. If anyone doesn't know yet, your name's Jamie. So Jamie, thank you for the introduction there. Please, everybody check the description box and you'll find the channel to Studio 215 official. I will be updating the thumbnail and the title, obviously, to reflect what this show actually is. And to the creationists out there, we're definitely going to want you to share this around because, again, it's an argument that has been utilized a lot over the last several years. And So I think it's a really great thing that we're going to have a comprehensive response to it. Jamie, I know a thing or two about having endless projects. And you and I were talking about before going live that we are hoping to introduce a new series at some point in the future, the Creationist Experience. And so we'll have several hosts, me, Jamie, maybe a guest creationist every time, and it'll be a call-in show where those of differing views and opinions can jump in and have a discussion with us. So, with that
so now that you've announced it, I just want to say, now that you've announced it, it's official. Like, we have to do it now.
Right.
So
the Creationist Experience is now official. So Jamie, again, I appreciate that. And to the audience, we don't know how the second hour will be. So feel free to tag me with some questions for Jamie. As you're listening to the presentation, take some notes. If there's some questions of clarification or objections, tag me, and we can also incorporate some Q&A after his presentation. But that being said, the link is posted. So anybody who is in the chat that disagrees with Jamie and his presentation here, click the link. I'll leave you backstage, of course, until the presentation's over, and then I'll bring you in and we'll have some impromptu debate. Okay, Jamie, we're going to hand it over to you, my good man. And whenever you're ready, let's get that presentation going.
All right. So this is kind of something we already saw in the first debate. I took a lot of the stuff from the first presentation, just kind of added to it. If we have time towards the end, I'll pull up the peer-reviewed paper that Taylor and a lot of the other evolutionists like to use, and we can actually just read straight through that. But I don't know if we'll have time for that, because I do have a feeling that there's going to be someone that wants to join in and, voice their objections. So we'll kind of see what happens with that on the second hour. But as far as this presentation goes, we're going to be talking about Chlamydomonas reinhardti. Here's the anatomy and micrograph comparison, so you can kind of get an idea. The reason I like to put this slide up is because they always like to say that, oh, this is a simple unicellular organism, but it's not simple at all. Like, there's a lot of systems that have to work in unison inside of this unicellular organism. So as we move forward, you may see a little green blob in the depictions on the peer-reviewed papers and on different slides that I have, but it's not just a little blob. There's a lot of things that have to function properly for this to survive. So I just wanted to make that clear. This is not a simple organism like they like to say all of the time. Chlamydomonas reinhardti is a single-celled algae that uses photosynthesis as its main source of energy. There are roughly 500 species of this. So within all of those 500 species of Chlamydomonas, keep in mind, it's Chlamydomonas is the actual algae, and then reinhardti is like the subversion of Chlamydomonas. So there's another one that's blue, there's another one that's red, there's another one that I think is yellow. So Chlamydomonas reinhardti is the one they like to focus on because that's the one that they use in the labs, but there's many different forms of Chlamydomonas. What's interesting about them is they can survive many different environments and can be found in fresh or brackish water, damp soil, lakes, ponds, saline water, the sea, and sometimes even airborne or in snow. So this organism can basically live anywhere, right? This is why this is a model organism. Well, actually, that's not why, but that's one of the reasons why, is because they can test it with all kinds of different environments. But the other reason is because it has a fast generation time. It grows in both light and dark climates, so that won't be affected in their research, and it can reproduce both sexually and asexually. So it's important to keep that in mind. The fact that it does both is an important piece of information to have, because as they present these peer-reviewed articles, they're always going to say that it's asexual, and they want to ignore the sexually, because when you look at the sexual characteristics of it, you'll see that it's very similar to what they're showing in these peer-reviewed papers. But we'll get into that as we get further in the presentation. So the ecological importance of Chlamydomonas. I felt like this was important to discuss. We're not going to go too deep into it, but essentially Chlamydomonas reinhardti is immensely important to humans and nature alike. It can be used as a natural food source, fertilizer, and so much more. In the water, it can do anything from feeding fish to helping the decomposition in lakes, oceans, and sewage plants. So it helps clean up our environment. On land, it is a great food source for a large variety of plants and animals because it is rich in vitamins, minerals, and nutrients. They actually tried to make it a food source for humans. I'm not sure who wants to eat algae, but they tried that. And what they realized was in the process of manipulating it to breed more, they actually made it to where it's not healthy for you. So for humans, it's probably not the best food source, but I mean, if you were out there starving, you probably could eat it naturally grown Chlamydomonas and you'd be all right. The most important benefit of this algae is the key role it plays in fertilizers, both in the gardens and forest, because it significantly increases the nitrogen in the soil. It's obvious that Chlamydomonas reinhardti plays a crucial role to keeping our ecosystems alive. Because of this, it's pretty obvious that God designed this algae in such a way that it can survive and thrive in just about any climate, no matter how ideal or harsh that climate may be. So now we're going to look at the reproduction process and as Kent likes to call it, making babies, right? So I doubt he's listening, but if anyone wants to shoot this over to him, let him know I gave him a shout out. So here's the methods of reproduction. There's the asexual, which is considered homophallic or homothallic. I think it's phallic, but that's how I pronounce it. And then there's sexual, which is heterophallic. So for asexual, zoospore, planospore, and palmella are like the most common. And then with sexual, you have isogamy, anisogamy, and oogamy. What's funny about that is when I made this presentation on Kent's channel and in the first debate, there was someone that said, oh, you forgot fusion. But for whoever that is, if you're watching, I want you to know that I did not forget fusion because the sexual phases, the heterophallic phases, that is the fusion. So I'm actually going into more detail on the fusion sexual reproduction phases. So I just wanted to point that out because it kind of bothered me. I never got to respond to that guy, whoever that was. Now, for reproduction conditions, if living conditions are suitable, then it usually reproduces asexually. So in normal conditions, you will see it reproducing asexually. If living conditions are unfavorable, then it can also reproduce sexually. Sometimes it'll still reproduce asexually, but it can switch over to sexually whenever it feels like it needs to. And that's usually when the conditions are unfavorable. As far as the asexual, I'm going to show a graph and I'm going to try and read the steps of this process, but I don't know how well that's going to go because on the presentation view, the next slide is kind of small. So we'll see how that works out. Here's the zoospore phase, and this is the graph that we're looking at for the process of a zoospore phase. So you get the visual. I feel like people really understand things better when they see the visual. But as far as the steps, the protoplast contracts and gets separated from the cell wall. Then the parent cell loses flagella and the neuromotor apparatus disappears. The protoplasm divides, forming two daughter protoplasts. Sometimes the protoplast may divide into 8, 16, or 32 daughter protoplasts. Each daughter cell develops cell walls, flagella, and transforms into zoospore. Then they are liberated from the parent cell by gelatinization. I think I'm pronouncing that right. Gelatinization just means it ruptures through the cell wall. So it's a big fancy word for saying that it actually breaks out of the cell wall, which is what you see in the fourth phase right before it goes back to the beginning. The zoospores are identical to the parent cell in structure, but smaller in size. Then the zoospores enlarge to become mature Chlamydomonas. Under favorable conditions, this can take place every 24 hours. So if you want the steps, there they are. Screenshot it or go back later to reference it if you really want to. But I'm just kind of reading through all of this while you look at the visual so you can visually see it as I'm explaining it. Then we go over to the aplanospore phase. And with the aplanospore phase, the aplanospores are usually formed under slightly unfavorable conditions. The protoplast rounds off and secretes a thin wall outside. During this stage, it does not develop 1, the parent cell, 2, the aplanospore formation, and 3, the hypnospore flagella. These non-motile structures are called aplanospores. At this stage, the aplanospore waits for more favorable conditions. So it's basically saying, hey, I need to hold up because I'm probably not going to survive if I complete the reproduction process yet. Then they will germinate directly or divide to produce the zoospores once the conditions become more favorable. However, in the event of harsh conditions, So we're talking about unfavorable conditions. But in the event of harsh conditions, it'll actually go to another step you see in the slides called the hypnospore phase. So here's the steps again if you want to screenshot that. But the reason I'm going through these reproduction processes is because all of the characteristics we see of these different reproduction processes, you're going to see them pretend that these are new novel characteristics, and they're not novel characteristics. These Chlamydomonas have been able to do all of this stuff from the beginning. However, as we're going to see in the peer-reviewed papers, what they're doing is they're artificially selecting it to add some steps in that aren't normally part of the reproduction process. So next we look at Palmella. This is a big one because this is the one that seems to replicate, or this is the one that seems to display what they're showing in the papers most often. And actually, in the paper that Taylor Snake likes to reference a lot, they actually try and make a distinction between the Palmella stage and what they're doing in this experiment. And there are differences to that. I'm not denying that. But at the end of the day, the differences don't really say that the differences between the Palmella stage and what they did in the experiments that are explained in the peer-reviewed papers, those differences aren't really new. I mean, we'll see those throughout all of these processes. It's just they manipulated it to where these processes now add new processes from other processes into them. I know that sounds confusing, but when we get to those slides, it'll make sense. So with the Palmella stage is formed under more unfavorable conditions like shortage of water, excess of salts, extreme weather, and so on. The protoplast of the parent cell divides to make many daughter protoplasts, but they do not form zoospores. The parent cell wall makes a mucus sheath around daughter protoplast, and the daughter protoplasts develop a gelatinous wall around themselves but do not develop flagella. These protoplast segments are called palmellospores. These protoplasts form an amorphous colony, and here's the key word that I want y'all to understand, an amorphous colony with indefinite numbers of spores. So in the peer-reviewed paper that Taylor likes to talk about a lot, it says that Palmella will only create a certain amount of spores. That's not true at all. If you look up the actual like textbook definition of this reproduction process, which is what I'm reading to you, this is straight from a textbook. It says that an amorphous colony with indefinite numbers of spores. So there is no actual limit to these spores. There is a kind of like a what's the word? A mean or the average spores, and that's kind of what people reference. Is they say, oh well, you know, this is normally what it does. But according to the textbooks, it can produce an indefinite number of spores, which means it can produce as many as it wants. There's no limit to that. However, normally it sticks within an average range. These protoplasts form a amorphous colony with indefinite numbers of spores, and it is called the Palmella stage. When favorable conditions return, the gelatinous wall is dissolved, palmellospores develop flagella, and the spores are released. Then they make new thalli, I can't pronounce that word, but you can read it when I get to the next slide, with either the zoospore or the aplanospore process. So from the Palmella stage, it'll either go into the zoospore process or the aplanospore process, depending on the environment, just to kind of keep it simple. And again, this is straight from the textbooks. It's explaining the process of this reproduction phase. So if you want to screenshot these or reference these, that's why I'm putting these slides up there, so you can go back and reference them if needed. So now we move over to the sexual reproduction phase. This is the heterophallic stage, or heterophallic stage, however you pronounce it. And first we have isogamy. If you look at the chart for isogamy, because whoever was saying that I forgot fusion, look at the chart. It actually says this is a fusion process. So just wanted to point that out. So with isogamy, the fusion of the first part is we have the fusion of gametes, which are similar in size, shape, and structure taking place. The two gametes merge for their anterior ends, or by their anterior ends, and fusion begins. The protoplast withdraw itself from the cell wall, then divide itself to produce 8, 16, 32, or even 64 daughter protoplasts. Each daughter protoplast develops flagellas and transforms into gametes. These gametes are liberated by breaking the wall, and then the flagella of the gametes secrete a hormone called gamone. Gamone is used to help gametes recognize the opposite strains. The wall at the place of contact dissolves and fertilization takes place, and a quadriflagellate zygote is formed. The zygote later on loses flagella and gets covered by a thick wall and is now called a zygospore. So there's one thing I want to point out about this process. Now, remember how I said that the gametes are liberated by breaking the wall, and then the flagella of the gametes secrete a hormone called gamone. Gamone's purpose is to help the gametes recognize the opposite strains. Now, not to take too much out of Kent Hovind's textbook, but how did that even evolve according to their model, right? So we have it's supposed to normally be an asexual process, but it also has a sexual process. So now evolutionists have to try and figure out, why does it have both? And how did it evolve this process of the hormone gamone? Because it never needed the gamone hormone in the asexual process. So now that it has something completely new in the sexual process, how did that evolve, right? It's just, there's so many complex things to this, what they call simple algae, that I don't think they understand that this is so much more than just a couple of little blobs of green algae multiplying, right? There's so much more to this than what they're wanting to represent it as. So again, if you want to look at the textbook terms and definitions, there it is. That's the whole process of the isogamy stage. I know there's going to be someone out there that's going to go through and try and dissect everything and say, oh, you forgot this step, or, you know, whatever their little argument's going to be. But this was straight from the textbook. So, I mean, if you're arguing with this, you're arguing with what the textbook says. Next, we have anisogamy. Now, with anisogamy, in the anisogamous reproduction, the gametes are unequal in size. So here's the thing. Isogamy is when they are equal in size, right? And then anisogamy is when they're different in size. So there will be a big one and a little one. That seems to be the major difference. So I don't know why they decided to make a whole separate reproduction process based on that, but I think there is a couple different phases if you look at the chart. So we'll talk about that in a second here when we go through these processes. The male gametes, which are going to be the small ones, the microgametes, are smaller. The female gametes, are the macrogametes, are larger. So the males are smaller, the females are larger. That's usually how it is in the animal kingdom. I know that algae is not an animal, but you'll see that throughout nature. Aside from humans, usually the female is the dominant of the species, or the kind, or the sort, or type, or whatever you want to call it, right? But it's really interesting that humans are the only ones where the male is the dominant of the species, kind, whatever. So I thought that was interesting. And, you know, that's another thing that we have to question. Why did that change in the evolution process, right? But neither here nor there. To get back on topic. So we have the male gametes, which are the microgametes, and the female gametes, which are the macrogametes. The macrogametes are formed in female gametangium, in which the protoplast divides to make 2 to 4 larger gametes. The microgametes are formed in male gameteum, or gametangium, where the protoplast divides to make 8 to 16 smaller gametes. The microgametes come close to this macrogamete. The protoplast of the macrogamete enters into the macrogamete, and after the fusion, a diploid zygote is formed. Now keep in mind, you hear that word diploid. That's going to be important because that's what they're going to use to say that this is, when they're doing their scientific research in the papers, you know, where they manipulate this to have extra steps in the life cycle, as they like to say, they're going to use this word, or they're going to use this phrase of simple multicellularity. And it has something to do, I actually wrote down the definition so I don't get the definitions confused, but it has something to do with diploids. So it's interesting that they say that this is novel, but yet we see that in the anisogamy stage, right? So it's not really novel. It's just they're ignoring the sexual reproduction part. Neither here nor there. The zygote secretes a thick wall and transforms into zygospores. After this, it goes through the isogamy stages for the zygospore. So what that means is, if you look at this chart, right, this is the process that we're going to see that's on the right side of this circle, right? So the result is either going to be the different anisogamy or isogamy stage. And then once they get to the middle where the zygote is in the bottom, then it's going to basically do the same process as it loops back around to the parent to create the parent, or to create the separate algae. Sorry, it's a lot of stuff that I'm trying to not overcomplicate, but in the process of simplifying it, it seems like I'm making it more complicated. All right, so here's the steps from the textbook if you want to, read that through. Now we're going to look at oogamy. Now, oogamy or oogamy, however you want to pronounce it, this one doesn't really happen very often, but it does happen, so we're going to talk about it. And the reason that I'm going through all the details of these processes is because the better you understand something, the easier it will be for you to notice when they're trying to do a sleight of hand. If you notice, evolutionists always do a sleight of hand. They'll say, well, we did this and this happened, so therefore it proves this, right? But they're not proving anything except for that they manipulated something that already existed. But if you don't know any of these processes and the steps of these processes, then you don't know that they already existed. So that's why we're going through the details of each one of these stages. So for oogamy, the vegetative thallus functioning as female cell withdraws its flagella and directly functions as non-motile macrogamete or an egg. The microgametes are formed by 4 divisions of protoplast, as in the case of anesogamous reproduction. The microgamete reaches the female gamete and unites by anterior ends, then the contact wall between the two dissolves. After plasmogamy and karyogamy, a diploid zygote, there's that word again, diploid, is formed, and then the zygote secretes a thick wall and becomes a zygospore. After this, it goes through the isogamy stages for the zygospore. So here's the steps for that. So as you can see already, there's a lot of functions that this algae can do in order to reproduce. And as we go through the peer-reviewed papers, if we make it there, that might be kind of the part that we're winging it on when we get to the second hour. As we go through the peer-reviewed papers, or if you decide to go read the peer-reviewed paper yourself, you'll see that all these functions they claim are new are not new if you compare all of these different sexual reproduction, asexual reproduction processes. So we're not seeing anything novel in these processes. What we're seeing is they artificially selected for clusters to make clusters as part of an obligate phase in the reproduction process. So this is an image of Chlamydomonas reinhardti during an ideal reproduction phase, right? So this would be more of the asexual phases. When it is less than ideal, it forms clusters, and usually it's under the form of aggregation. Aggregation is just a fancy way of saying it forms clusters under stress, okay? In harsh conditions, these clusters can build a wall around them to help them survive until conditions get better. Here's a chart showing some of the recorded social behaviors of Chlamydomonas reinhardti during stress. So if you look at this photo over here, this is the aggregate phase of reproduction, right? So if it goes under stress, it's going to form these clusters just like this. Now, in comparison, this is the pictures they use as figures in the peer-reviewed paper that Taylor and so many other evolutionists love to use, right? That little bottom corner square right there that my finger's on, that's what they say is a multicellular organism. They love to say it's not a cluster. It's not a cluster. Well, yes, it is a cluster, but we'll get to that. However, this is aggregation from stress, still single cell clusters. So on a completely different video that's just explaining the aggregation process, it shows the exact same thing that they're saying is a multicellular organism. So now let's define multicellularity, right? Because if they're going to argue that this went from unicell to multicell, well, now what exactly do you mean by multicellularity, right? From my studies, I've noticed that evolutionists have a very specific way that they want to define multicellularity in regards to this specific experiment. You'll see multiple different definitions, but it's kind of finely tuned when they start talking about Chlamydomonas. So first, we will look at and dissect how they want to define multicellularity, because they do it in such a way to fit their presupposed conclusions of evolution. Then we will look at the more accurate definition of multicellularity as defined by secular scientists whose main focus is to study science and not just push an agenda. When I say we're going to look at a more accurate definition, what I'm saying is I looked for a source that doesn't promote creation or evolution. So we're looking at a non-biased source explain multicellularity, and that'll be the more accurate definition that we look at. First, let's look at how evolutionists have decided they want to define multicellularity. And this comes directly from Taylor's slides from debate 1. They say that it's increase in size, cell differentiation, and functional organization of space. So we're going to dismantle all 3 of those. But this is quite vague for a definition of multicellularity, and there are a few issues with these characteristics in regards to Chlamydomonas and this peer-reviewed paper and studies and all of that they're trying to use as evidence. The first point I want to make is an increase in size alone does not mean anything. We see organisms increase in size all the time, and they never change into anything else. Remember, they're trying to prove evolution. So they're trying to prove that if enough of this happens, it'll create something else, right? But we're going to show you that that's not what happens at all. It doesn't even create anything new, but even if it did create something new, it's still an algae. So, not to mention that one single Chlamydomonas can reproduce up to 64 daughter cells in normal asexual and sexual reproduction phases. This is the average, but when you get to the palmello, all gloves are off. It can create as many as it wants. So, to see a cluster or colony of these Chlamydomonas extend beyond that is not unimaginable or a novel result, because that's the argument they're going to use. They're going to say, oh, it creates more than the average amount, so therefore this is novel. It's not novel, okay? Palmelloids can create as many as they want. They usually stick within an average range, but they can create as many as they want. So that argument's dismantled. For those that argue this isn't a cluster or colony, I will address that in just a moment. And if we get to the paper, if we have time to get to the paper, we will look at plenty of times where their own evolutionary scientists clearly define this as a cluster. So if you're saying it's not a cluster, they didn't say it's a cluster, then you didn't read the paper, because it's everywhere in that paper. I did a Control F, which is like a search inside of a PDF or a Word document. And when I Controlled F and searched for the word cluster inside of the peer-reviewed paper, I think it came in a total of like 64 times or something like that. So they clearly are calling this a cluster, regardless of what you want to believe. The second point I want to make is actually two-part. Dissecting the claim that we have witnessed cell differentiation in Chlamydomonas. That's the first thing we're going to focus on. They claim that they've witnessed cell differentiation, and we're going to totally dissect that. The second point that I'm going to make is that differentiation this differentiation proves evolution from single cell to multicell organism or multicellularity. They want to say that what they call differentiation proves this, but I'm going to dismantle both of those arguments. So this is a perfect example of cell differentiation. We have stem cells, right? And those stem cells will go on to differentiate into the muscle cells, into the fat cells, into the bone cells, blood cells, nervous cells, so on and so forth, right? But each one of those has a very distinct function for the body. If the body doesn't have the muscle cells, things are going to go wrong. If the body doesn't have the bone cells, things are going to go wrong, right? But these are very different from each other, right? They're not identical to each other at all. They're extremely different. You can literally look at them and tell the difference. This is not cell differentiation, okay? This is what they show and claim is cell differentiation. So I put blue dots and red dots on some of them because the reason they call this cell differentiation over here, the reason they call that cell differentiation is because the ones that are labeled red will be motile, which means they can move around, and germline. And then that means they can move around and they can reproduce babies. But the blue ones will be non-motile and somatic. So they're saying that because they've differentiated functions between each other, that's differentiation. But the thing is, that's not specialized differentiation. That's not specialization. When you look up the definition of cell differentiation, it's specialized functions of cells. So that's not it at all. The thing that I want to point out about this is actually the true definition of differentiation is the process in which a cell alters from one type of cell into a different cell with a specialized function. So this over here is not specialized functions because before they manipulated these artificially, all of these cells could do all of those things. There's nothing new that they're doing right now. However, when we look over here, this is specialized functions. The reason this is specialized functions is because only the muscle cells can do what the muscle cells do. Only the bone cells can do what the bone cells can do. And they're not going to be able to go back and forth between that. However, with this one over here, they can go back and forth. They've actually, in the article that Taylor likes to refer to a lot, when you look at the article and you read the part where it's actually giving the science that they did in the lab, there's multiple times where they say that these Chlamydomonas tried to revert back to their original state, but because of the centrifuge and the constant selective process, they struggled to revert back to their original state. And if we get to the paper, I'll show you that. So therefore, these guys right here are being manipulated into only doing one function and staying in clusters. However, given the opportunity, they want to go back to their original state. So that's not specialized function. So this specialized function consists of a new design. So go back over here. That's the same design, just they switched roles with each other. This is different design. The muscles have a completely different design than the bone cell. The bone cell has a completely different design than the sex cells. Okay. So this specialized function consists of a new design that did not previously exist prior to differentiation. So it's important that you see that last part. It did not exist prior to differentiation, because as we look at this chart over here, you'll see that the ones that are labeled red and the ones that are labeled blue, before what they call differentiation, they could all do the same thing. So therefore, they do not meet the definition of specialized function. So this definition is taken from a combination of secular sources such as Wikipedia, which I know is not fully, reliable, but bijust.com/biology. And that's a website that a lot of college students use for making notes and putting together research papers and stuff like that for projects. And study.com, which is another source that college students use to make notes for, papers, essays, all that. So 2 out of those 3 are very reliable. Wikipedia, not so much, but Wikipedia agrees with the other 2. So I put it in the list. All of these sources appear to mention nothing on either creation or evolution. Well, Wikipedia will definitely mention that. But on this specific definition that I looked up, they didn't mention anything on creation and evolution. So this is more of an accurate and non-biased definition of the term. Based on this definition, we did not see any true differentiation in any of these experiments. So if you go and read the paper, you'll see that according to the criteria for differentiation, they did not meet that criteria. Now, all that we witnessed in these experiments is artificial selection that took cells that were already prone to aggregation and encouraged them to form generational colonies, or as the papers say, clusters, that consist of daughter cells. Furthermore, the single cells within these colonies are being assigned roles that were already pre-existing in the DNA code. So all of the functions that they divvied among themselves in the cluster, inside of the cluster, some of them said, we're going to be the ones that are motile, and some of them said, we're going to be the ones that reproduce. Guess what? Both of them could already do that. That was already a part of the DNA code. So there's nothing new being put into that DNA code. In order for evolution to take place, you need new things being added constantly. And with the amount of generations they've done in these tests, we should expect to see something new, and we do not see something new. They're just adding one stage in that was not normally in that life cycle. However, it was in other life cycles of other sexual processes. Before the experiment, the single cells already had the ability to be both motile and germline. After the experiment, roughly half of them stopped being motile, and the other half stopped their germline function. There is nothing novel about this. It's essentially they artificially selected these to act as if they're in a survival survival environment. They have to form into these clusters to survive. They've artificially selected these to make these act as if that's what they're going through. Even though they're not going through any harsh environments in the lab, they are now acting like they are, which we see in harsh environments, they do the exact same thing. So again, nothing novel about that. So if these results prove anything, it's not that mutations throughout generations will, if these results prove anything, it's that mutations throughout generations will only cause a loss of function. What they're doing right here is they're losing functions. Some of them are losing the function to be germline, and some of them are losing the function to be motile. With that being said, these results really fit predictions made within the creation science model and is contradictory to evolution. So I think it's time that they give up this argument and, try and scrub these papers from their arsenal. Also, it's important to note that none of these functions are novel for two reasons. One, none of the functions listed are new to Chlamydomonas, as we've talked about already. And two, the life cycle behavior has been recorded in the wild. Remember how I said that they've seen this in harsh environments? Well, the harsh environment they saw that with is the Chlamydomonas that they call the snow algae variation. And it lives in polar regions. And it's also known as Chlamydomonas nivalis. Chlamydomonas nivalis in the wild actually does this. So nothing novel about that. They basically, in the lab, what they did was they took Chlamydomonas reinhardti and manipulated it to act like Chlamydomonas nivalis. So there you go. Finally, let's look at the claim that this is a functional organization of space. Because remember, Taylor and all of the evolutionists, they have those three criteria, and one of those criteria is that it's a functional organization of space. So this is a great example of a functional organization of space. Every single one of us is a great example of this, right? You have the heart, the lungs, and they're all in the right place to do their functions, right? And if they go out of those places, everything falls apart. That's a very functional organization of space. That is not a functional organization of space. That is a cluster. But by all means, don't take my word for it. Read your own papers. Here's a quote from their paper. It says, organisms that have never had a multicellular ancestor, they're talking about Chlamydomonas reinhardti, we subject the algae Chlamydomonas reinhardti to conditions that favor multicellularity, resulting in the evolution of a multicellular life cycle. Remember, not a multicellular organism, a multicellular life cycle, in which clusters reproduce via motile unicellular propagules. So there they are. That's one of the many times they admit that it's only a life cycle of multicellularity. It's not an actual multicellular organism. And as much as they want to say it's not a cluster, it's not a cluster, your scientists constantly say this in the papers that you're trying to reference. So get your facts straight. Now, let's look at a more accurate definition and characteristics of multicellularity. Like we did earlier, we will use the sources that mention neither creation or evolution to keep it completely unbiased. Multicellularity is organisms that are developed by cellular specialization and division of labor. Each cell becomes efficient in a single process and becomes dependent on the other cells to work in coordination for the proper functioning of the organism. At first glance, the evolutionists can try to say that this is what we see in their experiments, but when we look at the characteristics of a multicellular organism, it's obvious to see why this is not the case. Here are the characteristics of multicellularity. One, they are made of more than one cell, obviously. Two, they contain a membrane-bound structure, right? So they all have to be within one structure. Three, they have specialized systems for each other. Remember, specialized systems is muscles, bones, organs, so on and so forth. It's not one of them does this, one of them does this, but they're exactly the same and they can switch back and forth if we allow them. Four, they have distinct organs or differences in the cells. So as we showed with the human, we have very distinct organs, whereas with the Chlamydomonas clusters, they are identical. And in the paper, it actually says that if you allow them, they will revert back to their original functions, which is to do everything by themselves instead of rely on each other. Five, their cells exhibit a division of labor, which we can argue that that is part of what we see in those papers, right? Because I mean, there is a division of labor. Some of them decide to be motile, some of them decide to be germline. So yeah, there is a division of labor. I'll give them that one. And 6, their size increases with the number of cells. So yeah, we see that one too. But you're seeing 4 out of the 6 characteristics, and the 2 characteristics that you're missing are the most important characteristics for multicellularity. Given the non-biased, true scientific definition of multicellularity, the results of these experiments constantly being used as evidence do not meet the qualifications of true multicellularity. Not only that, but they are also lacking the 2 most important characteristics of a multicellular organism, which is specialized systems and distinct organs or significant differences in the cells. I have built somewhat of a passion for this remarkable creation, and I could probably talk about it all day long. However, I think this sufficiently makes the point that the peer-reviewed papers are trying to make in regards to nothing more than I'm sorry, let me re-say that because I lost myself on that one. It's a lot of words there. So I have built somewhat of a passion for this remarkable creation, and I could probably talk about it all day long. However, I think this sufficiently makes the point that the peer-reviewed papers are just a combination of intelligent input, which you can't have that for evolution, right? Because it came from nothing, not intelligence. Because if you add intelligent input, now you have God. But all we're seeing in these experiments is intelligent input and artificial selection creating pre-existing functions in Chlamydomonas and careful wording for the intention of deceiving, as is just about every piece of evidence that we see being used for evolution. So when they say that, oh, we don't really understand what they're saying in these papers, no, we understand. What we're really saying is that you're gullible and you're just taking their word for it and not even reading into what they're really saying. Because you'll see phrases in the peer-reviewed papers like, this happened 4.2 million years ago, and now we're trying to figure it out, right? And they make these dogmatic statements like, oh, it happened, so we have to figure it out. But then when you see the actual science being used, they openly admit, yeah, this is not a multicellular organism. This is not evidence for evolution. It's just a new life cycle or something around those lines. They openly will say that it's not evolution, but the way that they word it is what's really interesting, because instead of saying it's a multicellular organism, what they'll say is it's a multicellular life cycle. Or they'll say multicellular Chlamydomonas, ... whatever point they're trying to make. And then in the next sentence, these clusters. Oh, well, there you go. So look, they really are trying to word this in a way that makes it sound like they are multicellular, but they literally backtrack themselves in the very next sentence and say it's a cluster. So here's all my references. I'm sure y'all aren't going to be able to copy and paste that. So, you can just ignore all of that. But that's basically the end of my PowerPoint presentation. All right. I don't know where you're at, Donny, but if you want to bring in some people, I saw a lot of chats going out of the corner of my eye.
Jamie, excellent presentation, extremely comprehensive. And your slide where you showed a true multi-celled organism like a human being and in all of its complexity and sophistication versus the clumping that was observed in these Cladomonas experiments, I think was telling enough to say that the evolutionists really are grasping. Is that your ultimate opinion on this argument, that they're really just grasping for evidence for common descent?
Yeah, so I would say the common people like Taylor and James W. and all these guys, they are grasping for straws at anything they can possibly find that proves evolution. Whereas the people with the agenda, which is the scientists that are getting paid to prove this, they are intentionally lying. Like in their papers, they will say, like I said, there's parts in the paper where they'll say multicellular Chlamydomonas exhibits ... right? And then in the next sentence, they say these clusters. So hold on. Is it a multicellular organism or is it a cluster? That's the big thing you'll notice throughout this whole paper is they're constantly saying it's multicellular, but it's a cluster. And so no, it can't be both. It's one or the other. And so, yeah, there's gullible people that want anything they can possibly find to prove evolution. And they're taking it by trust, by faith, because it's a religion. But then when you actually dive into their religion and see the details, they're completely wrong.
So basically what I gathered from a lot of your presentation is, and I think you nailed it with the fact that these amoeba species are left functionally compromised because one group, correct me if I'm wrong, Jamie, one group in one of the studies was able to adapt to the introduction of a predator basically by cycling back and forth between the clumping and the free living state. Kind of like how an army comes together, an army of human beings in times of war, but in the absence of that threat of war,
they all go home.
Exactly. They all go home.
Well, so the paper that I'm focusing on is the one that actually doesn't use predation to do this. Because if you remember my debate with Luca, the debate I did with Luca, he used the paper on predation, but Taylor specifically tries to avoid the predation paper and focus on the one where they just use centrifuge, because then that means there's nothing being used to impress this change, right? But the thing that he doesn't notice is inside of that paper, it shows this process of centrifuge over the generations, and throughout this process, the single-celled Chlamydomonas are constantly trying to break out of this cluster. So the Chlamydomonas don't want to do this. So part of evolution, if I'm wrong on this, then feel free to correct me. But part of evolution is whatever is evolving, it has to want to do that, right? It has to feel that is beneficial to him or to them. However, these Chlamydomonas don't seem to agree with that. They're constantly trying to go back to their original state. And eventually they do go back to their original state. These obligate clusters that they're talking about, I know that's one of the main points that Taylor likes to talk about. He likes to say that, but they're obligate clusters now. Well, he doesn't say clusters, but he says they're forming this multicellular organism, he would say, but it's really a cluster. He's saying that they're forming that obligately, like every generation they do that. Well, yeah, but you artificially selected it to do that, and you haven't allowed it outside of the lab environment to see if it'll continue to do that. However, we see in the lab that throughout those generations, they do try and go back to their original state. Like you said, the army wants to go home. They don't want to stay in their unit.
Right. It would not be beneficial if an army were to stay in its unit, even outside of times of threat. But in this case, you have one group of algae that's able to transition from the clumping or clustering to free living. But in another study, they've lost the ability for that multifunctionality. And so overall, it's functionally compromising to the algae. There's no forward evolution. There's no large scale evolution. It's actually a loss of their pre-existing ability to adapt. Right.
Yeah, absolutely. A good way to explain it in layman's terms would be, let's say this army gets together, right? And some of them cut off their arms, and some of them cut off their legs. Well, now you have a compromised military, right? You want all of them to have all of their functions. So we're seeing a loss of function, not a gain of function. And that supports the creation model, which says, they're going to bring forth after their kind, and they're always going to do that. The more we try and change that, the more we're going to compromise them.
Well, according to our design diversity hypothesis, where God would have front-loaded the original created kinds, and that would include these algae variations with pre-existing and beneficial diversity, where environmental stressors and triggers can manifest the pre-existing ability to adapt. So all we're seeing here is, yes, phenotypic adaptation against predators, but that adaptation was pre-existing in the algae species, where you have one group of algae that's actually lost the ability to choose, depending on whether or not the predator is present in the environment. It can either cluster or it can go back to free living. But apparently, so Jamie, from your experience, the evolutionists, they look to the example of the algae that is now in a permanent clustering state, do they look to that example as essentially the stronger example for multi-cell evolution?
Yeah,
Because
well, the
now
thing
it's in
is
that permanent state.
yeah. So what they do is, because remember, in the paper it says that this is just a life cycle. This is part of its life cycle now. It doesn't mean that it's permanent. It's going to eventually go back to that single cell, and then it may go back and live in the clusters again. But yeah, that's essentially what they're doing, is they're focusing on this one part of the cycle and saying, there we go. We have a multicellular organism. It's never been an multicellular organism. It's still a cluster. And the paper constantly says that. I mean, in my opinion, Nature, the Nature peer-reviewed articles, those are probably the most reliable of all peer-reviewed articles. And they are clearly saying that it's still a cluster. So, I mean, you're arguing with your own people here, because they're trying to promote evolution, and at the same time, they're saying, no, this is a cluster. But one thing you said was about the adaptation. And what I think is interesting about that is, yes, God has designed us in a way to where we have front-loaded information. So if we go into a harsh environment, we can adjust to that. We can adapt. However, there's barriers to that adaptation, right? There's certain parts, there's certain layers that we won't get past based on God's limitation in our genetics. He limited us to do certain things. But what we see is that when the evolutionists try to pass those barriers, they actually are deteriorating, and they're making they're making whatever their creature is that they're modifying, they're making it weaker overall, because now they have to depend on each other. And then when they do actually disperse back into their own single cells, now they're not fully functional. So yeah, we see a loss of information, and the more we try and pass these barriers of limitations that God has instilled in our DNA, the more we actually see these negative effects.
Okay, so we have one group of algae that is expressing this clustering ability permanently. But in your more diverse populations of algae, are they observing this when being exposed to the predator? Are they observing this loss as well, where they're now permanently expressing the ability to cluster together? Do you know the answer to that, Jamie?
So I don't really. Can you word that again? I'm not exactly sure what you were asking there.
Well, because there's different groups of algae that they're looking at. One previous study, from my understanding, they were able to cluster together in the presence of the predator, but they were able to, once the predator was removed, they were able to go back to free living. And so they have that multifunctional ability.
Yeah.
But in the population that was now permanently express this ability to cluster, we're saying it looks like it's a loss of that multi-functionality. Is that a less genetically diverse population? Do you know?
Well, yeah, because some of them are now not able to go back to their original state. So yeah, they are relying on each other. So yeah, this is all we're seeing is loss of function. So the reason that they're sticking together is now they've been forced to go into these clustered states, and they can't function without it. Like I said in my debate with Luca, these single cells, they want to go back to their original state, but when they do so, they die. So that's when we see the end of that experiment. And then they just focus on another experiment where they're still in their state. But the thing is, they've never actually put them back in natural environments and just let them live, right? Just let them have their generations and see what happens. They're constantly keeping it under a controlled environment that fits the conditions of what they say favors multicellularity. What I'm willing to bet is that if they added the results of what happens when they stop putting them in those conditions that favor multicellularity, it will eventually revert back to the normal Chlamydomonas that doesn't rely on clusters to survive. So I think that's where we see a distinction between clusters and colonies, if I'm not mistaken, is colonies are now reliant on each other. So they're still individuals within the colony, but they're reliant on each other. However, clusters, they can rely on each other, or they can split off from each other. And I don't know if maybe that's wrong wording or not.
With a colony, it reminds me of a community coming together, and within that community, you have doctors, nurses, police officers, firemen, engineers. And so that community is all dependent upon each other, because if your doctors were to all die off, there's going to be a major problem there, because the doctors were the individuals that had that ability or expertise to basically function in the health of people, right? So they're but it's not like that community of individuals all became one unit, one human being. And so it's a colony,
Right.
but it's not an example of any kind of forward evolution. Where in the case of the clustering, I think you made a very powerful case that in your more diverse populations or groups of these algae, they have the ability to go to and fro from the clustering to free living. I mean, that is because they are more genetically diverse. There's more pre-existing diversity there to adapt. But in your one group that is now expressed permanently where it can only cluster, this is a loss of information, as you've put it. I'd probably say variability. So it's not actually taking things forward, because the algae, I'm sure, would love to go back to their multifunctional nature. So
Right. And see, that's what's interesting about that, is that's literally like what you just said. That's literally what they say in the title of this paper. Let me go and pull it up for a second so you can see that. Because it doesn't actually say that.
I was just going to say, so I've got this comment up here because we're live, so a lot of people are joining now. There was supposed to be a debate. We're rescheduling it because an emergency came up for Taylor. So our prayers are definitely with him and his family. And so within the next week or two, we are going to reschedule. So what we've done with this show is we've given a very comprehensive presentation. I thank Jamie for that. Almost an hour debunking the single cell to multi-cell argument. And now it's currently an open mic for as long as we can, specifically for evolutionists. So if there are any evolutionists that want to challenge anything mentioned or brought up in Jamie's presentation, click the link. It is pinned. Feel free to join. The last thing I want to say is we've organized an open mic tomorrow night as well with Dr. Dino himself, Evolution Challenge to all evolutionists. It is going to be hosted, though, on the Praise I Am YouTube channel. That is going to be at 8 o'clock EST, 7 p.m. Central. Okay, apologies, Jamie. Go ahead, brother.
Yeah, so what you were saying is that it switches back and forth. It can go to and fro. And a great way to explain that is the analogy you used with the city. That's kind of the exact same analogy I used in the first debate. That's pretty much the same thing I used. Just I think I used a family as an example instead. So yeah, we have the colony, which is, certain people can do certain things. And when one of those people is missing from the city, now you're missing a whole function of the city, right? That would be a good way to describe colony. But then you look at clusters. A good way to look at clusters would be the Amish, right? Now they live together, they function together. But if, let's say, one Amish decides he doesn't get along with the others, he can go branch off and survive on his own, 100%. He knows how to do all the farming, how to do all the stuff he needs to do to survive on his own. So that would be a good example of a cluster. But what's interesting about that is when you look at the clusters that are going on, and then we see that they keep calling it a cluster, but I think they might actually want to change that phrasing to a colony, because this is what we're seeing, is that eventually they get to a point where they can no longer separate back out and survive on their own. However, they haven't met all the conditions to be a multicellular organism. So they're stuck in this multicellular life cycle that would have eventually gone back to single cell. And what's interesting about that is that's literally what they say in the title of the peer-reviewed paper. So let me just full screen that for a second. And that one's backwards. Okay. So awesome. I forgot to swap that one over. Let me just transform that right quick. Okay. So the title literally says experimental evolution of an alternating uni and multicellular life cycle in Chlamydomonas reinhardti. So that's exactly what you just said. They're showing that it can express staying in a cluster or separating back out into its individuals. And they are openly admitting in the title directly that it will eventually go back to that unicellular life cycle and stay there if it's allowed. But the conditions that they put in, for whatever reason, I don't know what the centrifuge has to do with that, but something about the centrifuge that they're constantly doing to these generations and then selecting the ones that are more prone to staying inside of these clusters. Eventually they get to this point of selection where now these Chlamydomonas feel like they're obligated to stay inside of these clusters. So it's kind of like they're tricking the Chlamydomonas into thinking that they need to stay in these clusters to survive. And then the Chlamydomonas get to the point where they do need to stay in these clusters to survive. So you're seeing less reliability per individual.
Right. Basically, even the fact that they're using the word expression, a shift to the expression of these genes that pre-exist in the genomes of these algae, prove that this ability is already pre-existing. There's nothing new. From my understanding, these papers aren't actually providing evidence on how these novelties truly arose. I mean, what arose is differential expression, which means the ability is already there within the system. And what could have happened with your less diverse species of algae is they could have been hit with a deleterious mutation in their adaptive response to the predator, which left them without their multifunctional ability. And so now they are permanently stuck in this clustered state, where then if they were to go back into the wild, they would be weaker. They would be less built to adapt to the wild because they now have less genetic variability to adapt. So, I mean, as you put it, brother, in the presentation, they really do need to drop this line of argumentation, don't they?
Yeah, because so the argument sounds nice on the surface.
Yeah.
But then when you actually start picking it apart and looking at sentence by sentence in their articles, you see that it's actually stating the opposite of what they're wanting it to say. And there was another point I wanted to make. I kind of lost it there for a second. But regardless, as you read this paper, they say that, there's a new function happening, but there's not. And that's why I showed all of the reproductive processes. Because when you look at the reproductive processes that we know happen in the wild, it's essentially doing that in the lab, but they're taking they're making it do it outside of its original design. So what we see is artificial selection and mutations that actually leave it weaker. So yeah, they need in order to prove evolution, they need to not have intelligent input first off. And that's one of the things I love about Dr. James Tour is he loves breaking that down, that no matter what you do, you're putting intelligent input into the equation. So how do we prove this without intelligent input, right? So they're putting intelligent input, they're artificially manipulating, and then the mutations actually make the unicellular organisms in these clusters weaker. In order to prove evolution, they need it to be stronger, smarter, more resilient, and that's not what we're seeing.
So basically, we see a kind of pseudo multicellularity, and it's not true multicellularity by any means. And it's due to the differential expression of genes that are already present, because we already know just through simple epigenetics, environmental stimuli, that there are millions and millions of genetic switches in especially higher life forms that are just waiting to be turned on or off via environment. And so the question is, how did those genes come about in the first place? If they're pre-existing, where did those genes come from? How did they evolve? And I guess my question to you, Jamie, is do we know from these papers, do the authors of these papers tell us exactly what genes were involved in this kind of pseudo multi-celled evolution according to them? I would guess not.
So I'm currently going through the process of highlighting like word for word all the points that they're trying to make in this paper. And from what I've seen so far, they don't mention really much about the genes. I think they mentioned a couple of genes once or twice in that specific paper that I showed you, but they're just showing how it's reacting. They're not showing, is it a new gene? Was it there before the experiment? Is it now something that wasn't there before the experiment? And now, we see this gene emerging. They don't ever mention anything like that. And that's kind of what we want to see if we were going to prove evolution.
I would say these papers, they with the algae, they don't provide us interesting evidence for evolution, but what they do provide is an interesting starting point for us to continue researching how the environment triggers pre-existing genes to now express differentially, giving us novel adaptations. That's interesting. And that points to design, because that points to forward thinking. These are forward thinking mechanisms in the genomes of biological organisms that point us right back to who? The forward thinker.
Right. So that was the point that I think I was trying to make earlier, but I forgot about, is that all of these intelligent things we see functioning inside of this simple-celled algae, this is intelligence, right? So where did this intelligence come from? And to go back to that point, if you have intelligence, you have to have an intelligent designer, or someone had to input that intelligence. It doesn't just come from nowhere. It's kind of like how Kent says, you can throw a whole bunch of letters from a Scrabble box down on the ground. It's never going to make a sentence, right? You need someone to go in there and make the sentence. So, yeah.
Well, it's never going to answer the question of net gain versus net loss. And this kind of brings me to Lenski's experiments. It's something similar. So Lenski's long-term E. coli experiment, where he's had generations after generations, thousands of generations of these E. coli. And although there's been a few interesting adaptations observed, his populations of E. coli, they've shrunk in functional genome size. They've lost a lot of genes. They're now handicapped. If you were to throw them back into a natural environment rather than the artificial world created by Richard Lenski, they'd be dead on arrival. And so bacteria, algae, they seem to have the ability to lose information or lose variability in order to adapt in the short term. It's kind of like, I like using the analogy of a car. If I wanted to get better gas mileage, Jamie, temporarily, what could I do? I could remove weight from the car, right? Remove the seats, remove the front
rear view mirror,
the mirrors on the side, just start destroying the car. And I've now got better gas mileage temporarily, but it's actually destroying the car. It's not improving the car. It's worse for the car. And that's what we see in now your first group of genetically diverse algae. Okay. They have this really cool adaptation, pre-existing ability where, okay, we have a predator is introduced to the environment, and now they can cluster together and they can defend against this predator. Once the predator is removed, boom, they go back to free living. It's this amazing pre-existing ability to do this. Then your other group, they actually cluster, but then they lose the ability to go back to the free living state. And so now they're functionally compromised. So your second group is actually just an example of reductive evolution. That's all we ever see is reductive evolution, and not forward evolution, brother.
Yeah, it's like Kent says, if you lose and lose and lose enough, you'll eventually gain something, right? Like that's kind of the way they want to look at it. And a perfect example of how that doesn't work is look at American Congress, right? The more they borrow money, they're not getting richer.
Right.
So there was one thing I wanted to point out, though, because we were talking about how, this is really just evidence of variations, right? And the way that the scientific community is designed is so focused on proving this agenda, because there's other things that tie into evolution that we're not going to get into today. But they're so focused on making us believe this agenda that they're actually hindering real science. Now, all of these evolutionary papers and research that they've done, all the money they spent on this, if we were to remove evolution from the equation and just call it the study of variations, we could do some real good with that. We can see, what would thrive better in which environments. How can we adjust the environment to do better for these organisms? There could be a lot of good that comes from this if we were to just remove the evolution aspect of these studies. But they're so focused on, proving evolution that they're not using the data for what could be beneficial to people. They're using it for what could be beneficial to the agenda. And that's where we see a hindrance in science.
Right. Well, even today in some of your higher end cars, you have these built-in sensors, you have automatic braking, you have a backup camera where if you're backing up too close to another car, it'll start beeping. You have these safety features where if you're driving down the highway, but you start to venture off into the other lane, it's called lane control. It's pre-existing in the vehicle. It's designed to bring you back into the lane and driving straight again. And so this is an example of forward thinking with human engineers, but this is also what we see in algae. This is what we see in all biological life, this pre-existing ability to adapt. So this is amazing, more amazing reasons for us to study God's creation and to discover just how environment does trigger this pre-existing diversity that was designed in the first place by God, that was front-loaded in the first place. So it really does just go back, as you've put it, brother, to the designer. It goes back to God. It goes back to the forward thinker. Now, we've kind of addressed this, but I do have a couple questions here. This one comes in from Redefine Living. Sam said, this environment triggers the evolutionist.
Yeah.
So the evolutionists get an adaptive response to your presentation where they get triggered. It bothers them, Jamie, because they so badly want to believe that they, as a human, are related to a strawberry and these algae. You know, that's science fiction. That's not science. It doesn't belong in a science textbook, that's for sure.
Yeah, what's funny about that is throughout this whole presentation, you know, they're gonna what the evolutionists are gonna do, and I can guarantee you that we'll see some videos tomorrow in regards to this. The evolutionists are gonna ignore all the valid arguments I made, and instead they're gonna like hyper focus on the parts where I stumbled over a sentence in the slide, or you know, where I said colony instead of cluster or something around those lines. They'll focus on all of those little, you know, like public speaking issues, because I'm not that great at public speaking, so that's why I stumble sometimes. But they'll focus on all of that and completely ignore the actual arguments that are being made, because it really does trigger them, and they don't know how else to respond to it except for to go back to these papers that we're showing these arguments from, right?
Well, the perfect verse for those evolutionists that want to nitpick rather than, and you're a great public speaker and presenter, firstly, Jamie, you did an excellent job. But the evolutionists that want to ignore, it's a red herring. Those that want to ignore the bigger picture, the bigger differentiating arguments, and just focus on the minutia that don't really make a difference to the point. The perfect verse for them is Matthew 23:24. Ye blind guides, which strain at a gnat and swallow a camel.
Yeah.
And so rather than focusing on the minutia and just coming up with excuses, focus on the bigger picture. We put out an article. I appreciate it. Doki Doki. Christopher Roop, author of Contested Bones, and I, we worked for weeks. We worked diligently on this article, technical article, Genetic Entropy, refuting the critics. And so please, if you haven't yet read that article or check it out, do check it out. And it's one of those things where so far we haven't had any sophisticated response. But to genetic entropy in general, what you will find is exactly that, is these evolutionists, like your Dan Stern Cardinals of the world, they'll focus on the minutia while missing key arguments. And this actually brings me to genetic entropy in a way, Jamie, because they have this greater problem to solve. The accumulation of deleterious mutations that are putting shelf lives on genomes, them pointing to these examples of amoeba adapting to the introduction of a predator in a specific environment is not going to solve the degeneration process. So in the bigger picture, the bigger scheme of things for creation versus evolution, it does nothing. It does absolutely nothing to help them. So they're just stuck between a rock and a hard place. But I could rant about that all day. Let me get Redefine Living's question back up here for you, brother. And so he's asking, does this process create new information, and or does new information create this process? Or does this process occur with existing information? So Jamie, go ahead.
So yeah, when we did the presentation, you notice how I went through all the different forms of reproduction. And that's because I wanted to show you that Chlamydomonas can do everything they're claiming is new, all by itself in the wild. Chlamydomonas nivalis, for example, the polar version of Chlamydomonas, that one can literally do exactly what they're demonstrating in the lab. So that's what I was saying, was that what they did in the lab was they took Chlamydomonas reinhardti and basically made it act like Chlamydomonas nivalis. So that shows us that there really is no new information being added or coming from this process. What we're seeing is existing information being triggered. And I'm not that great with genetics, but I remember I watched a video that you and Matt did, where y'all were talking about, the front loaded genes and how they trigger on and off. And you use the analogy of a light switch, right? Like you flip it on and then, you don't need the light no more, flip it off, right? Like that's exactly what we're seeing here. However, in this case, they've flipped it on in a way that now if they flip it off, the light bulb blows up.
Right.
Right.
And now it's permanently off.
Yeah.
Or your alarm clock has a benefit because it wakes you up every morning for work or for whatever you got to be up for, right? And so that annoying noise, I guess, is not as annoying as it was back in the day now that we have our cell phones. But if something were to happen to your alarm clock where it's just permanently on, it's got that annoying noise all the time, right? I mean, yeah, it was beneficial when when it could go between the alarm and the not alarm. Mm-hmm. But now it's just permanently stuck in one state, which is what we see with the amoeba. So let me bring in, I think we're going to start wrapping it up here. We are at the 1.5 hour mark. I've had the join link in the entire time. No evolutionists have joined. But the challenge is out there. Jamie has the challenge out there. He's studied this thoroughly. He's studied this comprehensively. And so anybody on the opposite side that wants to debate Jamie on this topic, reach out. We'll set up the debate. Here's an 1.5 hour video, Jamie, where we are discussing the topic. There's a presentation. And so your arguments are out there. We will be rescheduling the debate with Snake Was Right, of course, sooner rather than later, Jamie, probably shortly after your James W. Debate coming up that I'm looking forward to, creation versus evolution. So with that, Jungle Jargon, good to have you, brother. What are your thoughts overall on the topic? What are your thoughts on what's been said? And what are your thoughts on Jamie's presentation tonight?
Yeah, I liked Jamie's presentation about the Chlamydomonas being restricted, but I think the evolutionists need to reconsider what they think as novel function, because restricted function, such as the case that they're trying to show us, is not a novel function because they're restricting the function of whatever it is they're trying to group together, Chlamydomonas, whatever. But I liked his presentation, but I do have a question. It's a little bit maybe unrelated, but I would like to know if anybody has been able to tackle the SNP haplogroups markers for that they use to claim an out of Africa descendancy. Is anybody ready to tackle that, or?
Well, for the audience sake, for Jamie's sake, SMP is a SNP, stands for single nucleotide polymorphism. The answer, Jungle, in short, is no. There are some very devastating problems to the evolutionists who want to still hold to this out of Africa theory. You're right, it is kind of off topic, so we don't want to get into that here. We want to wrap this up and keep it solely on the single cell to multi-cell evolution topic, just so people can share this around. And I want this to be a go-to video for people who are confronted with this argument from the critics. And so they can come to this video and it can be all things amoeba, single cell to multi-cell, Cladomonas, Ryan Hardy, and all that fun stuff. So,
I do have one question about that, though. So for the people that the people that stick to this argument of the whole out of Africa theory, I mean, what do they have to say about the whole traced study from Nathaniel Jensen? Like, do they just ignore that completely, or like how does that work? Because there's a lot of solid studies out there that show that like it clearly did not come out of Africa. So it's like, what how do they justify that? You know what I mean?
Well, that's a good study, but it's based on the rate of mutations. It's not based on the SNPs. It's based on the rate of mutations, because our mutation rate is way too fast for it to be millions of years, and that is a good study. But the SNP SNPs, as they like to call them, it's a single nucleotide polymorphism, which they use to link the families trying to claim an out of Africa descendancy, because the they link the Semitic populations with the Hamitic population by the way of C, which is the CF, and then they link the Semitic populations with the Eurasian populations, which is by way of the IJK. But it's an interesting study. I think I understand it pretty good, but I was just wondering if anybody has been able to address that, because to me that was like a big problem for me, because those are real genetic markers that link the different families. And the way I understand it is that it's not a marker being gained going left to right. It's a marker being lost from right to left, because you have to root the tree with the oldest haplogroup, the oldest Y chromosome tree, which would be the Eurasians, which would be the Japhethites.
Well, Jungle, I'm just gonna I'm gonna stop you. Great topic. What we'll do is probably another show where we can really get into out of Africa. We can get into trace. We can get into these SNPs. But I don't want to, as much as it's a good topic, I do want to keep this specific video just on this topic. And so, JJ, I appreciate you joining, brother. To the chat, I really appreciate everybody's input, feedback, and questions. I do find it funny, right, Jamie, that this, for a while, kind of like ERVs, endogenous retroviruses, have been one of the favorite arguments from the evolutionists. Mm-hmm. But the question is, where are the evolutionists tonight?
That's exactly what I was about to ask you.
That just got demolished. So
I was literally just about to say, like, that was going to be the next thing I said. Is I was going to say, I think this is a first in SFT history, because usually you go to that chat room and it's like flooded with evolutionists. And I think I've been scrolling through this chat for like the past like 2 minutes, and I don't think I've seen one evolutionist that has something to say, you know? So yeah, I think that's a first.
I like to say it's a great time. Oh, go ahead, Jungle.
The weather here is bad. The weather here is bad. I just wanted to be a place marker for, in case nobody else wanted to join. I just want to be a place marker, waiting for somebody to join. But go ahead.
I appreciate it, Jungle. Always a pleasure. You're very knowledgeable on these topics, and I appreciate your input. So what I like to say is it's a great time to be a biblical creationist, because their so-called best lines of evidence have actually turned out to be some of the best lines of evidence for special creation and separate ancestry. Endogenous retroviruses, not good evidence for evolution, amazing evidence for the design hypothesis. This argument here with the Cladomonas, these algae variations, amazing evidence for forward thinking with these pre-existing abilities to adapt. Pseudogenes, junk DNA, chromosome 2 fusion, you name it. All of their favorite lines of evidence have actually been overturned. They're actually some of the best lines of evidence for special creation. So as we wind it down, we are at the hour-and-a-half mark. I think this is a good time to wrap it up. And so I'm definitely going to share this around. I'm going to change the title. And there's really nothing out there. Jamie, correct me if I'm wrong. There's a couple articles out there on some of the Creation Ministries websites, but there's not really any comprehensive videos out there. And these days, people love videos, people love podcasts, people love presentations. That's what I listen to all day, right? Whether it's a lecture or podcast, debates. And so I'm really happy with this. I think it was meant to be, because we're still going to get the debate anyways. But hey, we got an hour-and-a-half video just absolutely dismantling this line of argumentation. And so now the next time it's brought up on Facebook, if it's brought up on YouTube, what people can do is track down this video and share it to your favorite critic. So, Jamie, I want to hand it to you, brother. Final words, final thoughts, and we'll have you kind of close us down here.
Yeah, I think that you were right. There's really not, like, I've heard a couple of creationist organizations that touch the topic, but I think they're kind of afraid to really dive into it. But I don't see why, because when you actually go in there and start looking at these papers, it's pretty obvious what's being shown. So yeah, as far as like my research on this, it has not been easy because there's really nowhere for me to reference as a creation starting point. So I'm essentially going in, looking at exactly what the evolutionists are saying, and then kind of like brainstorming, like, okay, what are they really meaning here? Because they're saying something, but it doesn't show that. Another thing I wanted to mention is every one of these arguments that they think is like solid evidence for evolution, it always backfires on them. And we see this over and over again, right? 100 years ago, it was these fossils that prove monkeys became humans or apes became humans, whatever they want to say. But then those got proven wrong. So then they jumped to the next thing and the next thing. And every single time they got proven wrong. And it goes through this cycle that reminds me of what Albert Einstein said. And it's like the definition of insanity is trying the same thing and expecting different results. They're trying to prove this theory, and every time it gets proven wrong, instead of tossing the theory, they toss the data that proved it wrong, right? And so that's kind of what drives me nuts about the evolutionists is like it's just like, when are they finally going to give in and give God the glory? And that gets to the final point that I wanted to make. And it's like, anytime you try and go up against God, you're going to lose. Okay. God is the all-knowing creator of all of this complicated and magnificent world. Right. And the thing is, I actually talk about this in one of my Bible studies that, human is constantly trying to understand the universe. They're constantly trying to figure out this great mystery of the universe. Right. But the thing is, the more that you try and dissect it and get further away from God, the less you actually understand. And in the end, you're only making yourself look like a fool, and you're rejecting solid understandings to essentially stick to what's already been proven wrong. So God's always going to win. And the way that I like to tell people this is, man is still learning, and we're always being proven wrong throughout this learning process. God doesn't have to learn. He knows all of this, and he's revealing it to us in a drip feed manner, right? Like, okay, you understand this now. Now here's a little bit more on top of that. But the thing is, God has never been proven wrong. As much as they try to prove God wrong, he has never been proven wrong. Man has constantly been proven wrong time and time again. So at some point, you have to look at this dilemma and say, do I want to side with the people that are always wrong, or also known as the losing team? Or do I want to side with the all-knowing creator that's ready to give me all the answers I need? Just like you said, we're at this point where it's a great time to be a creationist because all these things are coming to light, and the level of technology we have now is allowing us to finally explain God's creation in the manner that shows it's God's creation. So for anyone out there that's on the fence, now is the time to switch over to the winning team.
Amen. Amen. Great final words. You said it perfectly. To the audience, stay tuned for the predestined creationist experience show with Jamie, myself, and other creationists. Tomorrow night, again, open mic with Dr. Dino. Jamie, hopefully you can join us. Jungle, you as well. It will be hosted over on the Praise I Am YouTube channel. And so, real quick, I guess, toleration. You can find Dr. Dino on the channel Genesis Baptist Church, and he will be here
and
doing
also
some
Dr.
debate.
Dino.
And Dr. Dino as well.
Yeah, he has two channels now.
Amen. Centurion, thank you for joining. Says, is this live? This is live. It has been live. Please go back. Anybody just joining us, make sure that you go back right to the beginning. Watch it a few times. There's a lot to digest, as Doki Doki, my brother, has said in the live chat. You comprehensively debunked this favorite argument by the evolutionist. So Jamie, excellent job. Jungle Jargon, thank you so much for joining, brother. Always a pleasure. I pray you're doing well. Did you want any final words, final thoughts, Jungle?
Well, not really. Just come visit me on my live stream if you want. Sometimes, once in a while, I have a live stream. That's it.
All right. Well, everybody check out Jungle Jargon. He's doing some great work for the cause of young earth creation. Make sure to check out Studio 215 Official. And Jamie, you'll be back here in a week debating James W. So I'm looking forward to that. Okay, everybody. God bless. Share this content around. If you're not yet subscribed, make sure to hit that subscribe button. God bless all.